Nucleic acids. Nucleic acids are large molecules, polymers, consisting of carbon, hydrogen, oxygen, nitrogen, and phosphorus. They store genetic information and instruct the cell in creating proteins. There are two types of nucleic acids: deoxyribonucleic acid, which is DNA, and ribonucleic acid, RNA. The three main types of RNA are messenger RNA, or mRNA, transfer RNA, or tRNA, and ribosomal RNA, or rRNA. Nucleic acids are composed of chains of nucleotides, monomers. Nucleotides are made up of a sugar, a nitrogenous base, and a phosphate. Here is a diagram of the nucleic acid DNA from Richard Feynman's The Feynman Lectures on Physics, first published in 1963. The two chains of nucleotides are shown. A more contemporary diagram is on the next slide. This DNA diagram shows more of the bond structure and how the two chains of nucleotides are formed in opposite directions. The bonds and nitrogenous bases between the two sugar-phosphate backbones are also shown. More to follow. The structure and function of DNA will be discussed in greater detail later in the course. RNA is similar to DNA, but only has a single chain of nucleotides, nucleobases. It also has a different sugar and one different base. The bonds between nucleotides are called phosphodiester bonds. Like bonds between monosaccharides, they are formed by dehydration synthesis reactions. The carbon atoms are numbered, and the 3 carbon of one sugar bonds to the 5 carbon of the adjacent sugar through a phosphate group. RNA contains the sugar ribose, while DNA contains the sugar deoxyribose. Here is the difference. The sugar on one nucleotide covalently bonds with the phosphate on the adjacent nucleotide to form a link in the nucleic acid molecule. The nitrogen bases attach to a base on the other chain with hydrogen bonds. The bonds are key to the operation of DNA. The strong covalent bonds in the sugar-phosphate backbone create a strong molecule. The weak hydrogen bonds allow the DNA molecule to be split, copied, and then put together, which will be described later. DNA contains the bases adenine, guanine, cytosine, and thymine, A, G, C, and T. RNA contains the bases adenine, guanine, cytosine, and uracil, A, G, C, and U. The difference is that DNA uses thymine rather than uracil. Nitrogen-containing bases composed of 2-ringed structures are known as purines. Nitrogen-containing bases composed of 1-ring structures are known as pyrimidines. DNA is double-stranded. It forms a helix due to the angles of the strong sugar-phosphate bonds. Hence, it is called a double helix. Weak hydrogen bonding between nucleotides occurs, but in DNA it is between guanine and cytosine, and between adenine and thymine. Each strand is unique due to its sequence of bases. In this way, genetic information is stored in the sequence of nucleotides. Since the bases are not part of the sugar or the phosphodiester bond, the base sequence is independent of the backbone. Any base sequence is possible. Instead of nucleotides being attracted to other bases in the same strand, to create shapes, they bond to complementary nucleotides in a second strand to create the double-stranded helix.